intensive course which will cover a lot of, or I would say most aspects of somatic NGS and molecular genetic analysis in cancer, which is done in contemporary precision oncology. And it is not only for genetic counselors and oncologists, but also for pathologists, clinicians, and molecular geneticists. And along with me, Dr. Kumar Prabhash, who is a professor and head of medical oncology, very experienced, and he also has an experience of lab, not only doing precision oncology, but also understanding the genes and reporting. And along with that, a lot of faculty, who have experience in analyzing, in interpreting, and in advising patients about precision oncology based on this. And precision oncology is not always not something very new. Tamoxifen, it targets the estrogen receptor. And much before we knew about molecular genetic analysis, and even before IHC, most breast cancers are ER positive. And this drug was basically a targeted therapy. Of course, now we have moved to a different level. And this course is basically both for trainee and practicing genetic counselors, DM medical and pediatric oncology students, and medical genetic students. And we have, I'll show you the numbers. MD radiation or surgical or gynae oncology, then practicing medical oncologist, surgical and geneticist, and of course PhD and post MSc or post PhD genomic scientist. We have about 2 hours ago 1,338 people who had registered, and in them about 65 trainee genetic counselors and 70 practicing genetic counselors. DM medical oncology students 132, and practicing medical oncologist about 70, and then of course a whole list of others. The learning objective of the course basically is more to explain the concepts and also give an overview. And here the focus is on somatic. Of course, germline also we'll talk about, but we do different courses for germline. Here we will talk about somatic and also germline when it is relevant, and the alterations that drive the cancers and metastasis, not just any alteration. We'll talk about the driver mutations and actionable mutations, and how these variants are detected by either NGS or other molecular testing approaches, either on the tumor or in liquid biopsy. What are the clinical indications of NGS in contemporary precision oncology practice? What is the sample requirement for that? It's very important. The clinician should know, the pathologist should know. What is the workflow of the NGS and how the variants are interpreted and reported? And those who are coming from the germline background, for them, they have to understand the databases and the resources for interpreting somatic variants are very different. Many genetic counselors are not familiar with this. We will also discuss different methods to identify actionable genetic alterations, including fusion and expression, because the difference in the cost, the sample requirement, and the time taken. And in our seminars, we have 12 seminars covering each site. We will also discuss how germline and somatic testing complement each other in certain cancer and specific clinical contexts, and what are the counseling implications of somatic test findings and the therapeutic implication of germline test findings. And then Professor Kumar Prabhash, in the last lecture, he will talk about, that's the concept and the practice. There were the challenges in Indian practice and what things that our medical oncologists in Tata Hospital and other hospitals are doing to overcome these challenges. So for this particular lecture, which I'm going to talk about, I will talk about, first of all, cancer is a genetic disease, whether it is sporadic or genetic. Germline, distinction between germline and somatic variants. Gene structure and function, the central dogma of molecular biology. And then talk of oncogenes and tumor suppressor genes, gain of function, loss of function, and the precision oncology approaches for both these kinds of mutations differently. What is actionable, what is driver, what is passenger mutations. Knudson's two-hit hypothesis also I will explain because that has relevance. And then multistep multistep carcinogenesis, Vogelstein hypothesis and his model. What are the hallmarks of cancer? And some of the defects in DNA repair genes and mismatch repair gene, I will talk about tumor mutation burden and MSI and how it is studied, because that is, these things. And then rest in the remaining 8 lectures, we will go, everything will be one by one, all aspects that will be covered. So first of all, cancer, we know it is a dysregulation of the most fundamental biological process, that is the cell replication and its control. And this will not happen with any minor changes in the cell wall or mitochondria. This requires a very major change in the genes which control cell replication and its control. And factors which govern cell replication and integrity of cellular phenotype, they can either suppress or accelerate development or progression of cancer. So this is very crucial because cancer will not just happen just like that. So all cancers are a genetic disease, and I'm saying that irrespective of whether hereditary or not, because genetic alterations either in the genes or at a chromosomal level, they are a hallmark of a cancer cell. There has never been a cancer cell where the defect is only in the cell membrane or a mitochondria. There has to have a genetic alterations. But when we say cancerous genetic disease, we are referring to a somatic genetic disorder because a cancer cell has to have a genetic alteration. And in 5 to 10% of these cases, the starting point is a germline genetic disorder. That means they are born with that genetic alteration, and later on more alterations are required for the cell to become cancerous. So this is the concept of somatic. Soma, that means our body tissues, and germline, that means the germ cells which are there, which form the body. So somatic and germline, and cancer, both are important. In other hereditary conditions, we look at only germline variants, and in cancer, we have to look at both somatic as well as germline variants. And they're two different classes of gene. And of course, some genes have a dual function. They could also be tumor suppressed as well as oncogene. I will talk about some of those things. And it is not just genetic. Majority of it is genetic, but we know that some of the changes are also epigenetic, because epigenetic changes can influence the regulation of the gene or the expression of the gene. And ultimately, how the gene is expressed, that is really what matters. So that also, we are not going to focus much of that in this particular course, but just mentioning it. So for a few months ago, I just looked at various papers and how what has happened in the last 50 years. Discovery of the key cancer-associated genes, and I'm sure each one of you will be familiar with some of these genes, if not many of these genes. So it all started with the oncogene. So tumor suppressor genes came much later. If you see 1970 and 76, the oncogenes were discovered, and these were the two gentlemen who got Nobel Prize in medicine, Michael Bishop and Harold Warmus, and both of them have come to here in Mumbai, and they are both of them are Indophiles, and they discovered this. So Harold Warmus was a postdoc in Michael Bishop's lab, and they discovered these oncogenes, and very soon Sir David Lane found this TP53 as a tumor suppressor gene, could also have a dominant effect. And then later on, David Malkin from Toronto Children's Hospital in Toronto showed that it is the cause for Li-Fraumeni syndrome. Then, of course, there are many genes. And if you see, initially, most of these were oncogenes. Finding oncogenes was easier, and because they're gain of function, and you can show that there's an effect in what is happening because they transform easily. And then after that, many tumor suppressor genes as well as oncogenes, they were shown in different cancers, and they were shown to be used for precision oncology. Now, to get an understanding, and why I'm talking about that, because sometimes they're very basic things and we have not sort of paid attention, or we have some sort of fuzzy idea about it. So I'm starting with the whole DNA. We know humans have 23 pair of chromosomes, 22 autosome pairs and a sex chromosome. And DNA is all wrapped into this, and I won't go into details, but important that 98% of this is non-coding DNA. And I remember many years ago, we used to call it junk DNA. I mean, I think that was our ignorance that we called it junk DNA. Now, missing heritability or missing explanation of many genetic conditions, we know the secret probably lies in the DNA that we don't sequence, or we sequence, but we don't interrogate because we do not know what it does. So that is a problem. Of course, how we started sequencing, that the DNA, then of course came Dr. Sanger, and he told us, okay, how to sequence. So we do sequencing. And of course, now in the NGS sequencing, the title of our course itself says NGS. Again, going back to the basics, it's very important that the central dogma. So in this 3% of the DNA, which is the coding DNA, there also it's a specific stretch or sequence of gene that or DNA that we call a gene. So it has sort of enhancer, promoter region, very regulatory sequence, the exons, the red ones, and the introns. And finally, the exons, through the process of splicing, you get mRNA, pre-mRNA, mature mRNA, all these exons joined together, and the cDNA, and then you have a protein. And then protein also undergoes a post-translational modification, and sometimes that is also very important. So there's a specific structure of a sequence of DNA that is considered as a gene, and that is then translated, and then post-translational modification. Finally, it is the protein. So okay. So what finally we are looking in context of a disease. We are not evolutionary biologists. We are not basic biologists. We are talking in terms of a disease. We find a genetic alteration through sequencing. So which genetic alteration has caused this cancer? And just because we can sequence entire DNA, do we always get answer? And the reason is because 98%, 97% of the DNA that we have in these chromosomes, we do not know a lot about it, how they function. Again, something which very basic, which is the degeneracy or redundancy. So multiple 3 base pair codons combinations for each amino acids, because we have 4 nucleotides. So there will be multiple combinations which can cause code for the same amino acid. So this allows synonymous variant, which is that means a nucleotide has changed, but the amino acid has not changed. This would allow us to have a start codon as well as a stop codon, and also a missense variant. That means it has sensed some other amino acid, and instead of proline, you get a histidine or an arginine, or serine. Instead of serine, you get threonine. So this is the reason why we get, and of course, although we are called amino acids, so they could be acidic, they could be basic, they could be polar, they could be non-polar, they have a different structure. And finally, when the protein is made, protein has a shape, is a definite structure, and that is what is causing either development or growth or a disease. When we look at alterations, I'll broadly classify it into, one is starting with chromosomal aberrations, so which could be either numerical or structural. Any chromosomal abnormality or alteration would have some effect. Sometimes the phenotypic effect is mild, sometimes it causes a Down syndrome, and sometimes you can see it can cause a very severe phenotypic effect or a disease that we know that. We are not getting into this right now. For precision oncology, that is not so important, but one should be aware. Then large exonic deletions or duplications or insertion. That means a part of a gene, that means few exons or introns, they are duplicated, deleted, or are inserted. So invariably, if it is an important gene, imagine there are some 100 amino acids are added to that or removed from that. The protein's shape and structure will go for a toss. It will not be able to function normally. Then you could have a small insertion or deletions, what we call indels. It could be in-frame, that means just one or more amino acids are missing or added. It may or may not always cause a disease, because if suppose it is in the loop, it might just cause a little kink there, but it may not be in the protein-protein interaction domain or any other biologically important domain of that protein, so it may not have. But if it is a frame shift, that means it affects the reading frame and it all moves. It is only a matter of, it's a probability after 5, 10, 15, 20, bases or codons, amino acids down the line, it will introduce a stop codon. So you will have a truncated protein. So anything which is written in red font here, that is more likely to be disease causing. Anything which is in the green, it is less likely to be disease causing. Now, if you have a nucleotide has changed, but the amino acid is same, that means a silent or a synonymous. So invariably, it will not cause a disease. I would say 99.9% it will not cause a disease, and it will be a polymorphism or a single nucleotide polymorphism. And why did not I say 100% times? Because if suppose that happens in the first base of the exon, So although the amino acid has not changed, but it could affect splicing or the last base of the exon. So some of them could introduce either cause splicing defect or the expression of the gene. But by and large, they are silent. The name itself says silent. Missense, that means a different amino acid. And I think that's where when we'll talk about oncogene and tumor suppressor genes, in tumor suppressor genes, majority of these, it tolerates one amino acid or two amino acid changes from, say, alanine to threonine, it doesn't matter. Of course, in some genes, especially like P53 and some other genes, tumor suppressor genes, it could be pathogenic. But in oncogenes, these are generally missense variants, and they happen at a specific codon. I will talk about that. And the remaining of them are a variant of uncertain significance. Then the splice site variants, which affect the splicing. Again, I'm not getting into the details of this. They cause aberrant splicing of the protein. Almost all are pathogenic. And especially in a tumor suppressor gene, as I mentioned that, because if, intron could be retained or an exon could be skipped, the protein will completely be, go for a toss, it will not function. And a nonsense, that means it cannot sense, and it introduces sense a amino acid and rather it senses a stop codon, it will result in a truncated protein again, which will not be able to function. So why I'm describing these germline variants here, because for somatic variant description, how they are classified, that there will be a separate lecture. I'm just basically three categories, basically five, like those who are clinicians know, whether it is radiology, whether it is cytology, we call it 1, 2, 3, 4, 5. 4 and 5 is most likely it will be what it is, disease causing or a disease. And 1 and 2 are most likely not, whether it is BIRADS, whether it is many other conditions. Same way for the variants. So 4 and 5 are likely pathogenic and pathogenic, benign and likely benign, and 1 and 2, and B versus 3. And pathogenic depend on the type of mutation, which I explained to that. Truncating splice site, large genomic rearrangement, or a known pathogenic or likely pathogenic missense, which has previously been categorized. And because if it is in the germline setting, it should be very rare in the population genomic databases like NOMAD. Right now, we have the version 4 or 1,000 genome or several other population databases. And it should either never be there or extremely rare as a homozygous in population databases. So otherwise, you can't. The only exception is the founder mutation. Sometimes some founder mutations are very common in a community. It may be present in 1 in 50 or 1 in 300, 1 in 500 people, like the Ashkenazi Jewish mutation. And benign or likely variants, they will generally be deep intronic. They could be synonymous and missense SNPs. They may or may not be rare, but definitely they may not be so rare or as homozygous in a healthy population. And people have done functional or family studies to show that they are not pathogenic, especially if they are rare, this thing. And anything which doesn't fit into that criteria, we put it into VUS. So although ACMG, American College of Medical Genetics and pathologists, they have a very detailed criteria. I'm just sort of summarizing it here. Now, why do we want to look at these genetic alterations? As I said, we are not doing research for curiosity. I'm talking on the clinical setting. This whole course is for clinical setting. So when we find something in the tumor, which is somatic, whether by NGS or by other method, that is usually for treatment, precision treatment or precision oncology. It could be targeted therapy, immunotherapy, or synthetic lethality approaches. Or it is germline, which is either by NGS or other methods on blood or mouthwash derived samples. Again, it is many of these are used for precision oncology, like BRCA mutation or a mismatch repair gene mutation, and also precision prevention, because instead of just saying everyone do this screening, it is not easy to do screening. So it is focused, and also cascade testing and prevention in the family. So this is a sort of a nutshell as to what whole thing about the cancer genes and hereditary. And I'm just covering some aspects of germline, because later on in the rest of the course, there'll be very little discussion on germline. It will be all focused on somatic. So, why we, based on the family history or age of onset or bilateral, we suspect genetic counseling, then genetic testing, and based on what we find, based on that, either in terms of treatment, precision oncology, or screening and prevention. So again, back to basics. And why I'm talking about this, because I have seen many, PhD students, post-PhD scientists, clinicians, get confused in something very basic, and that has a effect on your understanding. Imagine 10 years from now, you will have, today itself, a lot of patients come with reports which are almost like booklets. They would have 10, 15, 20 pages booklets, and on top there is a summary, and you can't just go by reading. So you have to understand what gene they're talking about. And some of the genes that we find, especially with exome and genome, they are not even classified what group of genes it is. Proto-oncogenes or a tumor suppressor gene. If it is found something which is like a truncating or something else, they will just report it. So proto-oncogene, that means they get only one allele is affected, whereas tumor suppressor genes, both alleles are affected because, chromosomes, one copy here, one copy there. And this is a gain of function gain of function mutation in proto-oncogenes, because the effect in the normal cellular function of the gene product is enhanced, whereas in tumor suppressor genes, it's an inactivating or loss of function, and it is reduced. Now, I will come in next few slides while what confusion this can cause. So you are calling it recessive. Proto-oncogenes are dominant because you need only one copy of it to be altered to have an effect, so dominant. Whereas tumor suppressor gene, both copies have to be inactivated for it to allow tumor to grow. So it should, by that criteria, it is recessive. So it causes a confusion, and I will talk about it later in the next few slides. So I use this picture of a car simulator. Most of us have learned driving on the road like that, but some people learn it on the simulator. So remember, car has an accelerator and two brakes. Sometimes you forget the handbrake is also important. So oncogene is like an accelerator. Imagine you press the accelerator and just get stuck there. There is a kink there or something else, and it just gets stuck there. That car will be out of control. No one will be able to control that car. And but brakes, if you are an expert driver, even if your foot brake has failed and you are not just around the turn or something else, then you may be able to, when the car slows, you change the gears and you can apply the handbrake. So tumor suppressor genes are like handbrakes. You have two copies of tumor suppressor genes, and when both have failed, then tumor progression happens. Whereas oncogene, just one copy is mutated, and not just anywhere. Like in the brake, thread is cut from anywhere, it sort of it loses its function, and you will not be able to apply brake. Whereas accelerator has to get stuck at a particular point where there's a kink, where there's a hole. So that is where it is a specific mutation. So in terms of the mutations, in oncogenes, mutations are at specific codons or their specific expression changes. That is that is there in oncogene. Whereas in the tumor suppressor genes, you will see that the mutation spectrum is very wide. It is across, suppose it has BRCA1 has 1863 amino acids and BRCA2 has more than 3000 amino acids. Mutation could be in any of these amino acids. Okay. So characterizing a variant in a tumor suppressor gene, which is not a truncating variant, is much more challenging. In oncogenes, there are specific codons and they are already known, and that is what we take into account as an activating or a gain of function mutation. And because of gain of function, you can target that. So mutations are actionable, and driver means that is one which is actually pushing that cancer. But when you do that, more and more genes you test, whether in the tumor or whether in the germline, you will find more and more variants. And I think making that distinction is not always easy unless until you have studied hundreds and thousands of similar kind of tumors, and you have already found a driver mutation, and you see that this another variant comes in different cases, and it has no relevance, no clear correlation, whether histopathological or a clinical behavior. And then you deduce that, okay, this happens in many tumors. This is a passenger mutation. Imagine if when a car is driving, if you have a accident because the brake had failed or a accelerator was stuck. After all, when the accident will happen, many things will be damaged. So these are like, that was not the cause of the accident, but that was the effect of the accident because of this tumor suppressors. So putting it in a nutshell, so some alteration, and I'm more focusing this is on the somatic variants now. Some alteration in the DNA or the expression of the gene that can cause cancer. It can make them more aggressive. It can confer treatment resistance. And most of these alterations now, they can be detected, but often with complex tests. This is a not very simple test, and it requires, various kinds of NGS. You decide, NGS, you decide which you make a library of which different genes and different parts of the genes. You sequence them on different platforms, and then you have a develop different bioinformatic pipelines to detect it, and then you say correlate. Unfortunately, today, only some of these alterations can be targeted. Like P53 is a very commonly mutated gene, but it is still not targeted. We can't really exploit that defect in the cancer cell. And even when we target, the results are variable. In some cancers, the results are remarkable. You get a very long, deep remission. In some cancers, you just get basically a few months of prolongation of survival. Sometimes even that much is also not seen. I'm coming back to the Knudson's hypothesis and retinoblastoma, and I'll explain to you why. Because tumor suppressor gene and oncogene is so it was well known that RB, retinoblastoma, which is always happens in children, it could be either familial or non-familial. So familial hereditary, non-familial sporadic. And so about 2/3 of these are sporadic, and most of these are unilateral. This was also well known. And about 1/3 of these are hereditary, and majority of hereditary retinoblastomas are bilateral. If they live long enough, they will develop retinoblastoma in the other eye. This white, for the people who are not doctors or oncologists, is white eye, what we call white eye reflex. It's a sign of a underlying retinoblastoma. In this unilateral one eye here, both the eyes are affected. So Alfred Knudson, what he did, he was a clinician, pediatrician. In 1971, he published a paper. He plotted, so bilateral cases, he assumed these are hereditary. These are the ages in month and what age they got retinoblastoma, and unilateral and bilateral. So very clearly you see the bilateral ones, and mind you, this is a log linear plot. This is a linear 10, 20, 30, 40, 50, 60 months. This is 0 months, 2 months, 4 months, 6 months. So this is a linear. So percentage of cases not yet diagnosed with, at the age indicated. So at birth, no one has retinoblastoma. And by those who have to develop retinoblastoma by age of 5 or 6 years, almost most of them do develop retinoblastoma. This is a straight line. This is a curved line. And those who have studied some forms of biology or radiobiology will know that when you see this kind of shape with a shoulder and then you straight line versus a straight line on a log linear plot, that means 2 events are happening here and only 1 event is happening here. And this is how he came up with the 2-hit hypothesis versus 1-hit hypothesis. And that was in 1971, he published this. And although he used retinoblastoma, what he found in this that was very seminal, and that explained relation between hereditary and sporadic cancers, even adult cancers, proposed a mechanism of penetrance in hereditary cancer. And in 1971, tumor suppressor genes and, both copies were not even, published. He predicted the existence of tumor suppressor genes with a very simple observation. And interesting, he wanted to become a mathematician, then became a doctor and pediatrician. So I think that little bit of mathematics, which he knew, just simple plot of 30, 40 cases in his hospital, he came up with this. So this is what I have so in a hereditary form, you are born with the first hit. Each cell of the body has the first hit. And then any one of those trillion retinal cells have to acquire a second hit. And once they acquire a second hit, then, they become transformed and then progression into this. And that's why, whereas in a sporadic form, all cells are normal at birth. And then the first hit happens in one cell and the same cell has to have a second hit. And that's why then that will proliferate. So as a result, because here you have advantage, so the cancers will develop at a younger age. They will be multifocal, multiple primary, and younger age because they have a lead time in origin. And many of these cancers which are driven by a germline variant as opposed to a somatic variant driver, they have organ specificity, specific IHC marker or molecular marker or a syndromic feature, depending which gene is involved. So why I'm showing this is, pattern of inheritance. We know that autosomal dominant, recessive, sex-linked, non-Mendelian. In some of our PhD, exams, I used to ask, okay, so so tumor suppressor genes, okay, what kind of syndrome they call, autosomal dominant or autosomal recessive, let us say RB or BRCA1. So they all used to write, it will cause autosomal recessive, inheritance. I said, why everyone is getting it wrong? It will cause autosomal recessive. Because at a cellular level, if you look at, where is that? At a cellular level, both copies have to be mutated in a tumor suppressor gene. So at a cellular level, it is recessive. But at the level of that person or that organism, that inheritance is dominant. Because if you are born, these are high penetrance genes. If you are born with one mutated copy in all cells of your body, there's a very high chance, like for retinoblastoma, more than 90% chance that in your lifetime you will develop one of more cells will develop a second hit and you will develop that cancer. So although at cellular level it is recessive, but as a syndrome, it is an autosomal dominant syndrome. Similarly, in BRCA1, Lynch syndrome, all those things. So at a cellular level, it is recessive for it to cause disease, but as a syndrome, it is a autosomal dominant, except some rare syndromes like CMMRD, where both copies of the mismatch repair genes are mutated, but then the form of that presentation of the syndrome changes completely. Instead of adult onset colorectal or endometrial cancer, they get childhood brain tumors, leukemias, etc. So, I'll, cover shortly because we started almost 25 minutes late because of the mix-up in the beginning. So Vogelstein, he proposed a model of colon cancer. That was because, that just talks about that, how you put it in a cancer. So what he proposed, Vogelstein, and I don't know who else was there in his lab. I'm forgetting the name of that person who was the first author in this paper, seminal paper. So a normal colonic epithelium, the APC gene is inactivated, and beta-actin activation, you get an early adenoma. So the model. Then you have a KRAS or a BRAF activation, intermediate adenoma, then SMAD4 or TGF beta 2 inactivation, late adenoma, P53, and then after a carcinoma is formed, then other mutation that leads to metastasis. So in this was a very simplistic model, and of course he used the FAP patient. They have multiple adenomas. He used that, and that was just to show that it's a combination of oncogenes and tumor suppressor genes, one after another. It will be very rare that all of them together they undergo genetic alteration. One after another in a sequence, they will undergo this thing. But finally, for a cancer to develop, there will be multiple alterations that would happen that will give rise to the cancer development. The exception is some of the cancers where just one, like BCR-ABL or something else, RET mutation, that just one alteration itself sets the triggers and that cancer would develop. So if I put it all together, malignant transformation is a multi-step process in which there are additional genetic lesions at each step, and that it's a cumulative effect which gives malignant phenotype to the cells. The transformed cells undergo clonal expansion, and subsequent genetic alterations results in metastatic or treatment-resistant phenotype. And that is where actually we become most interested, because ultimately patient wants that my cancer should be cured, it should not spread, and that's where we face a challenge. And I've already explained that oncogenes are proto-oncogenes which are activated due to mutation in specific domain, and they become like an accelerator of a car that is stuck in full throttle position, and driving cell replication is a crazy speed. But that is something you can actually relieve it also. You come and relieve, or you can block the effect of that. So that's why they are targetable. Not all oncogenes are targetable, but many are. Tumor suppressor genes are like natural brakes of the car, which fail when both alleles are made dysfunctional due to inactivating mutations. Okay, so except few tumor suppressor genes where could have a dominant negative or some other kind of this thing, mostly these are inactivating mutations in tumor suppressor gene. I am now introducing this. I will not get into the details of hallmarks of cancer, but I would suggest all of you will read. It will take you, reading, several hours of reading, then going through again. But finally, when a cancer grows, see, it's not easy to be a cancer and to sustain something that as a cancer for long. It needs a lot of things because it is fighting against the body, it is fighting against its own, it is growing so fast. So it develops several things, mechanisms, and most of these have a genetic basis, or some of these have epigenetic basis, like evading growth suppressors, avoiding immune destruction, enabling replicative immortality. That means it has to, continue, otherwise it will just die. Then tumor promoting inflammation, activating invasion and metastasis. It has to go to a different niche areas and then, grow there, induce or get access to vasculature because it needs blood supply, and genome instability and mutation, resist cell death, and, several other things. So, and there are many other emerging hallmarks and enabling characteristics, that is, we call enabling characteristics, that is non-mutational epigenetic reprogramming, polymorphic microbiomes. A lot of research is happening. We haven't sort of been able to fit it together, senescent cells, and unlocking the phenotypic plasticity. So this is just to show that it's a very complex system, just because sometimes we are lucky. That's why we said that it is not always easy to target driver mutation. Even if you target mutation, you may get variable results because it's a very complex scenario. It is not as simple as that. Now, I will just explain one thing about mismatch repair genes and microsatellite instability as an example, and then I will end my talk. So we know Lynch syndrome, it was earlier known as hereditary non-polyposis colon cancer, HNPCC, and this risk depends upon the age of the person, the sex, which is the gene mutated, and zygosity. So heterozygous mismatch repair mutations, they have Lynch syndrome associated cancer risk starts from the age 20 years. MLH1, MSH2 have the highest risk, 70 to 80%, most common colorectal. MSH2, PMS2 have a little lower risk. And extracolony cancers, the highest risk is with MSH2 and MLH1, and endometrial cancer risk is as high as 50% for endometrial cancer. Then a host of other cancers. But the same gene, if it is homozygous or a compound heterozygous, it gives rise to a completely different scenario, which is a childhood cancer predisposition. Almost all of them will develop cancer in, before they are teenagers, either brain tumors, leukemias, GIs, lymphomas. So what happens, I will just come into the how we look at it, because the concept for you to understand, looking at microsatellite instability, and this I will just, so 15% colorectals are dMMR, D means MMR deficient, or MSI high, I will explain that. So this has both prognostic and predictive value. And some of these are due to Lynch syndrome. They might predict, okay, there may be underlying Lynch syndrome or a germline mutation. And the phenotype of these tumors, they are mostly right-sided with poor differentiation, abundant tumor infiltrating lymphocytes. But majority of these, 2/3 are there, they are sporadic. That means they don't have an underlying germline mismatch mutation. And the sporadic ones, as compared to the germline ones, they have the same tumor phenotype. Okay, but they usually in older individuals, more in women, are in smokers. And in half of these patients who have mismatch repair dMMR, they have a specific mutation. BRAF is also oncogene. So, in oncogene, you write the codon and which amino acid is changed to which. Sometimes it doesn't matter that V600 could be changed to anything in some of them. So that specific mutation. And whenever you find this mutation, it suggests that there's no underlying germline mutation. That explains why this person got this mismatch repair deficient colon cancer. And you could have CIMP, phenotype, and silencing. Sometimes there could be a epigenetic silencing of the MLH1 gene. That can also happen, again, which I'm not getting into detail. So now I'm going to tell you about the microsatellite instability. So again, why I'm talking about this, I've seen many students, when you ask them, they know MSI, but you ask to explain them how it is done, they get sort of confused, can't explain. So I'm just spending few minutes to explain that. So, I can't say that she sells seashells by the seashore. If I'm saying slowly, maybe I can say it. If I have to say it again, maybe I'll make a mistake. But if I have to say it quickly, repeatedly, 5 or 6 times, I'm sure in the second or third attempt itself, I will mess it up. So this is, just imagine your normal cells, they have all the time, everything is going well for them, they are multiplying, so they get it right. But the tumor cells are always in a hurry. Imagine so many things. You look at the hallmarks, they're always in a rush, they're always in the hurry, they have to replicate also quickly. So in that process, they are more likely to make a mistake. So in that 3 billion bases, there are stretches of DNA, what we call is a microsatellite. So these are unique sequence repeat units. Okay, so they are repeat units, like you have said GGTA, GCO said AAA, so that there could be 20 times A mononucleotide. So 20 times T or G. Then dinucleotide CA, it could be N, means it could be 10 times, 15 times, 20 times. Trinucleotide, tetranucleotide, pentanucleotide. So these are repetitive sequences. So like when you have to she sells seashells by the seashore, that means that is a repetitive is coming again and again. So in the replication process, when you have to replicate very fast, there could be errors, slippage errors. So instead of say 20 times CA, it could become 21 times, 23 times, or 18 times. Okay. So for the patients or individuals who are born with mutation in, so I'm just showing this example, this thing. So this is a normal cell, which is a lymphocyte-derived DNA, which is the FFP tumor-derived DNA. And this is a NIH Bethesda panel of markers. So these are all, some of these are mononucleotides, 2 of these, others are dinucleotide markers. As I told you, lymphocytes are normal cells. They have all the time in the world to sort of replicate properly. And so they get it right. Whatever they inherited from their parents, the same, times of repeats. So what is done, you can see these peaks. This is the average length. So when we do fragment length analysis, after PCR, you run it on a fragment length analysis on a automated DNA sequencer, capillary sequencer. And you might find that the length of this is with markers, that size markers are there. It is 140 bases or 160 bases, 120 bases. In the tumor, if you look at, you can see 2 sort of set of peaks. One set of peaks, a second set of peak, whereas in the lymphocytes or the normal DNA, there was only one. You can see multiple peaks, but it is like this, this. So that means another species has come. Similarly, for this BAT26 in the blood and the tumor, you see that another species has come. So this has become changed. Okay. So what we call is microsatellite stable is when all the markers are stable between the lymphocyte derived or a normal tissue derived DNA and the tumor derived DNA. When MSI high, we call, then out of those 5 markers, at least 2 or more of them are unstable. And so that is considered, this will happen only when there's an underlying defect in the mismatch repair system. I did not introduce all the system, but I'm just saying. So this system, this is one of the DNA repair system. There are many DNA repair systems. You will later on hear about homologous recombination repair pathway and the defect. So mismatch repair, this is the type of thing when it happens in the body, it repairs. But when it is not functioning, it will not be able to repair. And MSI low, when only one marker is unstable, you are not really sure whether that is because of underlying gene mutation. So this is how you look at. So this is one way of predicting that this tumor is, something is defect in the mismatch repair pathway. Why it can happen, in the earlier slides I have shown you there are multiple mechanisms through which it can happen. Now you can also do by the immunohistochemistry, and I know there are a lot of pathologists in this. So immunohistochemistry, in this we look at these, you have to know which gene and protein. In immunohistochemistry, looking at the protein, where it is expressed, nuclear or cytoplasm or cell membrane. And you also have to have a control. So you have, in this, IHC is done antibodies again, MSH2, MLH1, PMS2, and MSH6. And the lymphocytes and normal colonic glandular epithelium, they serve as the internal positive control. And for all of them you run, and you see that if in controls it is expressed, so we look at this loss of nuclear expression. So if there's loss of nuclear expression of MSH2 and MSH6 in the tumor cells, where it is retained in the lymphocytes and the the rest of the normal epithelium, glandular epithelium, we know that this is loss of expression, nuclear expression, and this is MMR deficient and this thing. And in these cases, you may find that when you do germline testing, you might find there is a mutation. Generally, it will be a truncating mutation, germline mutation in either MSH2 and MSH6. I'm not explaining why MSH2 and MSH go together, MLH1 and PMS2. Just remember MLH1 and PMS2 together and the 2 MSH together. So that is how they pair. So again, I won't go into the details. So just one or two quick things. Last slide. So the prevalence of somatic, I'm talking of tumor mutation burden because of immunotherapy or a lot of things, these lingos, words are used, buzzwords. So in this paper that was more than 10 years ago, published in Nature, they looked at the tumor mutation burden, which is in the tumor, somatic mutation prevalence, number of mutations per mega basis. So there have been several studies, but I think by and large, the same findings are consistent. So you would see some tumor, like this is pilocytic astrocytoma, which has the lowest mutation burden of 0.04 MB and is driven by a single abnormality of the MAP kinase pathway. Okay, that is one. And again, rhabdoid sarcoma again has a very low this thing again because it was driven by a specific mutation in SMART-CB1 gene. But on the other hand, you see melanoma, lung cancer, bladder cancer, and most of these are because of the carcinogens. They have a very high mutation burden, and these are the ones where immunotherapy has, a lot of results you get, and because of high tumor mutation burden. And this is for the sporadic cancers, but in the germline mutation, if there's an underlying Lynch syndrome or a germline mutation, these tumors are also hypermutated. And if it is a CMMRD syndrome, that means both copies of that one of the mismatch repair gene is mutated. These are ultra hypermutated tumors. Okay, so yeah, I think this is our last slide I will talk about. This is for the germline. We have a huge care gap. So whenever in the next course, we will have a focus on germline genetics. So we need a lot of people who need to work on this. And our own, till about few months ago, we had done more than 8,500 germline NGS panel in our center, in our lab. And we found mutation in 31% of them and a lot of viewers. Oncogenes, I would want to say, I was telling you the phenotype may completely change. There's a red proto-oncogene depending where is the mutation. This one you see, this purple line, by the age of 20, almost everyone has got medullary thyroid cancer. Whereas in this one, light blue, by the age of 75, only 30%. That depends where the mutation. So M918, okay, in the M918, if that codon you have the mutation, you get MEN2B and a very, very high risk of cancer. These are all our own data. I think I will finish this. So this mother and daughter, those of you with the spot diagnosis, you can see she has these spots on her face. Her daughter has faint. And she went to a doctor. He thought that she had a retinoblastoma, removed the eye. Retinoblastoma, normally we don't do a biopsy because it will cause seeding. So directly do an enucleation. And it turned out to be a benign tumor. So this lady has tuberous sclerosis, and it was a tuberous sclerosis associated benign condition. So patient will come to a clinician and finally they will go back to clinician. In between, there could be a molecular geneticist, there may be a genetic counselor, there may be many people, there will be a pathologist. So clinicians, at least they need to remember the important aspects of medical genetics. And if they do not remember, the patients will not derive the maximum benefit of advances in molecular diagnosis and therapeutic. So like for this condition, there are mTOR inhibitors and many other conditions, many other things that you could do. But I think you have to suspect. A clinician, when they suspect, they have to know the context before they order for these complex and expensive molecular genetic analysis. Okay, I think with this, I will end my talk, and I'll again apologize for the late start because of the mix-up. So if there are questions, I will go to the YouTube. I'll see if there's any questions in that. I will address a few of the questions. 300 people are watching. Can any one of you see the question? I don't see any questions in the YouTube. If not, no. Is any of the faculty there want to say something? Okay, I'll stop sharing. Okay, so I think we will end the talk here.